Good afternoon, everyone. I'm Suganya Sivagurunathan, a postdoctoral associate at Beth Simoni's lab at Broad Institute of MIT and Harvard. With me today, Beth Simony and two other postdoctoral associates, Paula Janos and Shatavisha Dasgupta, will be joining us during the workshop, helping you during the workshop. Thank you all for signing up for the workshop to understanding how to unravel the hidden biology in your microscopic images. Let's get started. I will give an overview of how this workshop of these 2 hours is going to be. First, I'll be going to the overview of Morpheus, what it is, and how we'll be using to understand the hidden biology in your images. Then it will be followed by a demonstration. I'll be giving a demo on how to use Morpheus. And it will be followed by a hands-on exercise where TAs and Beth Simony will be helping on your questions. So to begin with, as we all know, with the introduction of digital cameras, every other image that we capture today is nothing but just an array of numbers. All the analysis that we do on the images are a bunch of mathematical operations performed on these array of numbers. Since each pixel has a ton of information hidden, there's a lot of information to dig from all the images that we have. In a typical quantitative microscopy experiment, a biologist tags a particular protein of interest, protein or organelle of interest, which they hypothesize might be altered upon perturbation. Let me quickly grab my laser pointer, so it's easier. Yeah. So a biologist tags a particular protein or organelle of interest, which they hypothesize might be altered upon the perturbation. And they image the particular tag protein or organelle and do measure the changes that is going on upon perturbation and comes up with the biological answers on what happens to these protein or organelle that have been tagged upon the perturbations. But in contrast, in an image-based profiling, we let the cells speak for themselves. What do I mean by that? Instead of focusing on one particular protein or organelle, we stain the entire cells, like all possible organelles or compartments inside a cell are stained using various dyes. And then with these, we'll be able to measure all the changes that are taking place inside the cells or the organelles upon the perturbations. Measuring these features, we'll be able to unravel new biology. We might unravel new biology that is going on inside the cells. By measuring features, I mean every other organelle or compartment that is being stained, we would segment those and measure probably features or texture, the granularity, or whatever you're interested in for answering a biological question. Let me dive into detail of how this is typically done. So this is a typical image-based profiling workflow where you culture cells on since it is generally done in a high throughput manner, we culture cells in a 384 or 96 well plate. It can be done on a smaller scale as well. We culture the cells and perturb them using either perturbing them with introducing siRNAs or expressing plasmids of different genes of interest, or you can even treat the cells with chemical compounds, or you can do a control versus disease sample cells as well. Once the perturbations are done, the cells are then stained with various possible dyes to reveal the features of different organelles or compartments inside the cell. I'll explain it with using a cell painting assay that we typically use for image-based profiling. In a typical cell painting assay, we use 6 different stains, which reveals details about 8 different constituents and organelles. Once we have the stained cells, we then use high throughput microscopes to image at various different channels, and we get a lot of images out of it. Once we have all those images out of it with each organelles or compartments of the cell stained, then we use a cell profiler. In that case, you can use any image analysis software to segment the objects of interest, and we measure a lot of features from this. Right now, cell profiler would give out nearly 5,000 measurements, 5,000 to 6,000 measurements, but you can also feel free to use other image-based image analysis softwares to get the measurements out. Once we have these measurements, a collection of measurements of a cell, like the measurements might be area, again, texture or granularity. A collection of such measurements of a cell is called as morphological profile or image-based profile. Once we have an image-based profile for a perturbed cell, then it is easy for us to compare whether the features of the perturbed cells are different from the control cells or not. This is what we call like image-based profiling, and we compare them. And then we can cluster in the downstream analysis to know if there is any similarity between these treatments or not. Having said all that, how do we actually look at all these measurements? These measurements are just numbers, but how do we associate? How do we look whether they are similar or not? This is where Morpheus comes in. In practice, we use something called similarity matrices in Morpheus, where we try to understand the similarity between the morphological profiles that you got out of measurements. I'll be talking about similarity matrices in a couple of slides, but before that, let me introduce you all to Morpheus. Morpheus is a free web-based software originally created by another group in Broad Institute for exploring mRNA profiling data. This software will also accept like CSVs and GCTs as input. So irrespective of whether it is an mRNA profiling data or image-based profiling, we can use Morpheus to explore large-scale associations of the data. And it allows matrix visualization, and also you can display various charts using this software. The main advantage of Morpheus is no extensive computational or statistical experience is required to use this software. And with the help of Morpheus, we will be able to understand the hidden biology in your microscopy images. The main applications of Morpheus would be to examine the correlations. Correlations in the sense, I mean the similarities between the samples to check for their replicability. You can also examine the correlations among the samples, and also if you find any interesting correlations or anti-correlations, you'll also be able to identify what are the features or what are the measurements that make these control cells and the perturbed cells different from each other, or why are they similar or why are they different. We can able to identify the features that make them similar or different. With this, we'll be able to understand the biological meaning behind all the measurements that we make out of the microscope images. Before we actually start using the Morpheus, there are a lot of steps that needs to be done with the data that we have. So the first one is the extraction of features, which I just shared about. But before moving on to before start to look at the similarity matrices, there are a lot of steps in between, which I'll go through one by one. So the first one is the extraction of features. This can be done with any image analysis software. We typically use Cell Profiler. And once we have the measurements of all the cells, we would be having measurements from each individual cell. The next step is to make average per well. I would explain it here. So we have got all the measurements from each individual cells. We don't. What we next do is take an average of all these measurements for each individual wells, right? Because it is easier to compare each well to every other well in your high throughput experiments rather than comparing millions of cells across each other. It is generally done it is generally easier to compare smaller data set than millions of cells compared to each other. And in another way, there's a lot of heterogeneity that is in a single well, where you have one of your cell might be going through a cell division phase and other one might be in a growing phase, but you want to have average of all the measurements that is taking place inside a single well. Having said that, with this tool, we'll be able to see only the major systemic changes that are taking place between two different wells and not the subtle ones. Once we have taken the average of measurements per well, what we then do is a normalization, like any other biological experiments. This technique also has a normalization step. The normalization step can be done either to all the data in the particular plate that you're analyzing, or it can be done to the negative controls. When to choose whether you want to normalize to negative controls or to the whole data. We have a blog post describing this, which I've linked in the end of the slide deck. Feel free to check it out. And why normalization is important. We have taken all the measurements out from the cells, but each of the measurements would be varying in a lot of different scales. For example, your cell's area might be in the range of 1,000 square pixels, whereas your intensity of the cells might be rescaled to 0 to 1. And there, if you're looking at the neighbors of your cells, that might range in between 30 to 50. So each of these measurements would vary so much that you don't want to be comparing all these across each other. So you want to make it into a uniform scale across all the measurements. I'll also demonstrate why normalization is important during the demonstration. And once that we have the normalized data, the next step we do is select a subset of features, which we call it as feature selection. Why do we want to do that? Although having me mention that we want to take a lot of measurements of outside the cell. The important feature, the important point, why do you want to do feature selection is to remove the redundant features. In the downstream analysis, when we are going to be making similarity matrices, it depends heavily on the features that are being inputted inside it. So we don't want to have a lot of information about the same feature to be inputted, which gives a lot of weightage to the single measurement, which is why we want to remove redundant features, which again, I'll be showing during the demonstration. Once we have all the average measurements, normalized and feature selected data, the next thing is to do a Pearson correlation coefficient. What do I mean by Pearson correlation coefficient in terms of image-based profiling? Pearson correlation coefficient is a way to show the measurement of similarity between two variables. In this case, we are trying to measure how similar one group of cell in a particular well to another group of cells in a particular well. So you want to see how different two wells are or how similar two wells are. So how do we do that? Since we have all the measurements from each individual wells, we plot all the features across two different wells and get a plot like these, where you can see the Pearson values to be like 0.9, which means a Pearson value of 0.1 means the two variables that you're comparing or the two wells that you're comparing are highly correlated to each other. And a value of 0 means no correlation, and a value of -1 means they are anti-correlated. So this is how you understand how similar two different wells are, how similar or dissimilar two wells are. So for now, we just got the comparison between two different wells. How do you do it on the whole plate? That is where similarity matrices kicks in. In similarity matrix, what we do is, the way we try to do a Pearson correlation between two different wells, we try to do across the whole plate or all pairs of columns or rows to assess the covariance and the features between these wells. So how this works is, let me point you to the similarity matrix here. We have the Pearson correlation coefficient from different columns and different rows, where you can see in the columns, you can see the metadata well, that is the well number, like different wells being plotted. And on the rows also, you can see metadata well, where you have like a lot of wells being plotted. Each square in the similarity matrix indicates the Pearson correlation coefficient between the two wells. So as you can see, a red square here between B2 and another B2. When you compare B2 to B2, it is highly similar because you're comparing same well. So you have a similarity matrix of 0.1. Similarity matrix of 1, which I'll be again showing during demo. But whatever is shown here is a similarity matrix between well C2 and B2. And since it has a value of 0.9, which means it is highly similar to each other. So when this Pearson correlation values are in positive, you see it is being colored in red. And when they are in negative, they are colored in blue. And the white means it is there's no correlation. So generally, the blue squares indicate anti-correlation and the red squares indicate they're highly correlated. Having said all that, like any other technique, this technique also has some caveats, which we need to be aware of before proceeding to or concluding biological conclusions with the image-based profiling data. The main caveat is, since we are making all the measurements based upon the segmentation and taking the features out, your segmentation needs to be good enough. The segmentation, it's not just necessary that your segmentation is good enough only in your control cells. It needs to be good enough across all conditions, like in your negative controls and also in positive controls and across all conditions. Because when you're making the image-based profiling, you don't want to see the differences caused because of your segmentation differences, but what you actually want to see is the differences that comes out of real biology that's going on. So how to know when your segmentation is good enough? We have a blog post for that, and I've linked the blog post at the end of the slide deck. feel free to check the blog post. And the other caveat that I would mention, it is always important to look at your image data and also your like the numbers data, both in the bioinformatics stage and also in the imaging stage. Because as you can see on the right side, there are 13 different plots which have similar mean, standard deviation, and correlation. But when you plot the X and Y, they look completely different to each other. So it is always better to have a look at your data both at image stage as well as the bioinformatics stage before coming to biological conclusions. So to conclude, morphological profiling can be a very powerful way to explore the cellular phenotype data. And you can measure lots of features of your cells or whatever structures you're interested in using Cell Profiler or any image analysis software that you would prefer. And by collapsing, doing a normalization and feature selection on your data, you'd be able to understand a lot more about the biology that's going on. And with Morpheus, we'll be able to do all these without the use of command line. Yes, and let me know if you have any questions so that I can proceed to the demonstration. If there is no questions, I will proceed to the demonstration. I posted the link to the Morpheus and also to the materials that will be useful for the workshop. So this is how a Morpheus layout looks like. Let me quickly grab. Okay, I think I'm not sharing the Morpheus screen. Sorry. I will just quickly share my screen again. Yeah, hope you're able to see it. Thank you. And yeah, this is how a Morpheus layout looks like. And I've even in the workshop materials, I've added the input data that you can use to explore the morphological profiles using this software. I'm going to be using one of those files. First, let me show you how is it. So these are the different measurements. I hope you're able to see the screen and with the Morpheus. And yeah, once we load in the data, what you are actually looking at is whatever the columns are the different wells, and the rows shows the different features that were measured from each well. So once you've loaded in, as I scroll through the different squares, you could see when it is cell area shape, which is indicated in here, you could see the different wells have average values to be in thousands, where you can see 4,000 or like it's all in 1,000 because it's area. But when you move your cursor to something like area shape or form factor, which actually is between 0 to 1, you could see all the wells are having values in the range of 0.5 or again 0.5 and like 0.56. So if you go to some other feature like probability perimeter, again, it's going to be something different. It's in hundreds. So each of these features that are on the rows are going to be different in their scales. That's why we want to do a normalization, because you want to have all the features in a close range than whatever we are seeing here. How do we do that? Once we have loaded in the data, we can go to Tools, choose Adjust, and choose Robust Z-score. Once that's done, now when I go back to the same area shape, you can see it's no longer in thousands, but it is in the range between like probably 1 and like -1 to probably +1. And if you go to form factor, of course, like it's going to be all within the same range. And again, perimeters, yeah. So now you can see that the whole range of measurements that we were seeing before were normalized to a smaller range. This is why we need to do normalization. And even in the exercise, you'll be playing around with the normalized and normalized data to see how that impacts the similarity matrices. So once we have the normalization done, we can further go and add a lot of features. Sorry that I indicated the columns are the different wells. We can understand by adding more annotations to the columns. How we can do that is there's a small gear symbol here on the top, which if you press that, it's the options. And if you go to annotations, you have row and column annotations. For the row, it is going to be the features that we have. But for the column, we have different annotations that is added, that is plate. You can add like plate, well, compound, or concentration. As you can see, when I keep adding these, there are a lot of details that are added on the columns. So with this, we'll be able to know which well these samples are from, or these measurements are from, which well and which plate, and what compound they were treated with, and what concentration they were treated with. So all these details can be understood by adding these features in the column annotations. Once this is done, I would now load probably, I will also show how to filter the features as I was mentioning about the feature selection. When you, to do that, if you go to tools and use filter, and we want to choose whether it is rows or columns. Since we want to filter the features, since features are in the rows, I'm going to be choosing rows, and you can click add. And if you want to see, yeah, since CP feature is nothing but a self-profiler feature name that we get out of the measurements. So if you choose CP feature name, you would have, we have measured the compartments of like cells, nuclei, and cytoplasm. If you want to see only the features from the nuclei, you can just type in nuclei here. And you can see all the, and there are like around 210 nuclei, 210 measurements that are obtained from nuclei. So if you want to do only, if you want to have a look at only the features from the nuclei, then you can choose this way and close, and you can see all the features from the nuclei are listed. Sorry, I'm going to use the filter again or go back to the raw data. And when I was talking about the feature selection, I mentioned that we need to give equal importance to all the feature measurements that you're making. Why did I say that? Let me quickly show you. So if you are interested in area, then probably you can see there are 144 different measurements that comes out from area. Whereas if you go to texture, is updated, and you can see there are like 340 measurements. So we don't want to be giving a lot of importance to the texture because there are a lot of measurements. We want to be giving equal importance to the different measurements that are made, so that the similarity matrices that are formed at the end uses each of these measurements giving an equal weightage rather than having a different weightage based upon the number of measurements that we have. This is why we want to do a feature selection. Both the normalization and the feature selection can be done using Morpheus, but in our typical workflow, we use a Python package called PyCytominer. But if you are not comfortable in using Python packages in command line, feel free to use Morpheus, but using the Python package would definitely give a better result than whatever you're doing with Morpheus. If you're interested in checking out the command line or PyCytominer, feel free to use the link at the end of my slide deck. But this is how you do feature selection and also do a normalization. Now let's go on and do a Pearson correlation coefficient and see how the similarity matrices look like. So for that, I'm going to be choosing an already feature selected data. I'll open the Morpheus and select file. And in the files that I shared, there's feature selected data, which I'm going to be using right now to generate similarity matrices. Once we open this, again, the columns are the wells. We can confirm that by adding more annotations to it. So the row annotations are the features again, which is already there, but I want to do add annotations for columns, where I'll be adding well, plate, concentration, compound, and MOA is nothing but the mechanism of actions of the compounds that the cells were treated with. So once I checked those, you could see that all the columns were filled with the details from the different wells, like the mechanism of action, compounds, concentration, wells, and the plate details as well. Once this is done, I will also show how to see if there are outlying features. Once you have, although you have done normalization and a feature selection, we can have a look at the outliers, how we can do that. Once you go to, again, go to options. Oh. 1 second. Seems there's a lag in the display. Sorry, let me. Okay, sorry, was it from feature selection or normalization? I would like to go over it again. Sorry for that inconvenience. Okay. Yeah, thanks. Sorry for the inconvenience. Seems there's a lag in the process that I do, and it was what it is being displayed on the Zoom. I would go over the feature selection again. What I basically did was when I chose the annotated data, I did normalization. How did I do feature selection? I chose tools and then use filter. Is it all going fine now?
Yes.
Yeah, thanks. Yeah. So once you choose filter, since we want the features to be filtered out, what I'm going to do is filter out the rows, because that's where the features are displayed. So the rows is chosen here and add. The field would be the CP feature name, because those are the CP feature is nothing but the features that we got out of Cell Profiler. So those are the different features that are displayed on the rows. If you want to select few features, probably I'll go over the feature selection again, because when you want to do area and hit enter, you can see there are like 144 measurements related to area. But if you want to do something else like texture, you can see there are like 340 measurements related to texture from the same plate. So it does not mean that we have a lot of we should be giving a lot of importance to the texture measurements over the area measurements. Since when we are going to be making similarity matrices, we want all the measurements to be given equal importance. So we need to make sure each of the measurements that we get out of whatever image analysis software that we have are given equal importance when we are making the similarity matrices. And this is why we want to do a feature selection. Generally, feature selection can be manually done using Morpheus, but in a typical workflow, we use a Python package called PyCytominer, which you can see the link at the end of the slide deck if you are comfortable using Python packages. Or you can give a first pass at Morpheus too. So this is how you do a feature selection. Like once that is done, if you just hit enter and close. So whatever the area, I think I selected the area, so you all see the features that are pertaining to the area alone. Now I will move on. I think you're seeing my screen still okay. Yeah, okay. And I'll move on to do a Pearson correlation on already feature selected data that I have. So once we load it in, again, we'll add more column annotations because we want to know which well the measurements are from. So I would go to column annotations, uncheck the ID, add details of well, plate, MOAs, the mechanism of action of the compounds that the cells were treated with, then compound, concentration. And I would just leave the row annotations like that because we already have the feature names there. So now we can see the columns have the details of the concentration, the compounds that they were treated with, mechanism of action of the drugs, and you can also see the plate details and also the well where the measurements are from. Once this is done, this data is already normalized and feature selected, but if we can still check if there are outliers. How do we check if there are outliers? You can go to options, like you can click on this options, and Morpheus generally uses a relative color scheme. It kind of decides the scales between the maximum and minimum measurements in each individual row. But when we uncheck the relative color scheme, it kind of does not do that anymore. Like there's no scale between the maximum and minimum measurements in each individual row. And then we can give minus 1,000 to 1,000 probably just to for a range. And when you're doing this way, you're now only seeing a portion of the matrix. We can use this zoom to fit to window where you can see the whole matrix. You're seeing it now blank because I think this batch does not have any outliers. That's why you're seeing it blank. If there's an outlier, you would see a bright red or bright blue line, which kind of indicates, although you've done normalization and feature selection, there are some outliers, which is a good way to know. And then we can either remove it or at least know if there's an outlier. Since we don't have any outliers, I'm going to be proceeding back to relative color scheme. And we can see this way. This is the whole, this is the measurements from the whole plate. Now we can go ahead and start doing a Pearson correlation between all the wells based on the large set of features. I would slightly zoom in so that you will be able to see the details of the compound and also the wells. Yeah, once you zoom in there. Yeah, before doing Pearson correlation coefficient, I would like to color all these details individually so that when we do a similarity matrix, we will get a better idea seeing the color. So we can right click each of these tabs, and then you can choose show color so that like wells that were treated with similar compounds would be colored similarly. Same way we can do it for other details as well. You can see when you color it by plate, all the plates that are from this, I mean, all the measurements from the same plate are like colored similarly. You can do that for concentration too. So once we have colored all those, and we can do for the wells, and well, I don't think it matters because anyway, I would color. Once those are colored, the next thing is doing a Pearson correlation. How we can do that? Go to tools and do a similarity matrix using Pearson correlation. And it can be done either on rows or columns. Since we are interested in understanding the similarity or differences between the different wells, I'm choosing columns here because we have the columns in the we have the wells in the columns. Yes, we have got the Pearson correlation and coefficient between all the wells across large set of features that we have measured. As you can see here, I will quickly zoom out so that you can understand the whole diagonal is going to be in red because each well is compared to the same well. So we have a Pearson correlation coefficient of 1, and that is why it would look red. So to zoom out, you can just click this zoom thing and fit to window. And you can see the whole diagonal is in red because it is the wells are compared to the same wells like each other. So it's just in red. But that's not our interest. We want to know what wells are similar and what is not. So I'm going to be zooming in again so that you can see the details of the wells and the compounds. Yeah. Put this one here too, so that you can see the row details as well. Yeah. Now when you see, I believe I could have zoom out. Okay. The well B2, whatever you see here, when we try to see to the, you can see the details of the columns here when it is highlighted. And if you click on this, and you can see the details of the other well that is being compared to. So when well B2 is compared against B2, it is the Pearson correlation is going to be 1. But you can also see the well C2 is also highly similar to B2, and well D2 is also highly similar to B2 because all of them are treated with DMSO and it is from the same plate. So this means like all the morphological measurements that comes out, or all the measurements or the morphological profiles of the cells from these wells are highly similar, or they have similar cellular phenotypes because they were treated with just DMSO. And as you can scroll through, you can see there's a big chunk of red here, which says like every other well that every other measurement that comes out from these wells were treated with DMSO, and that's why like they're all like in the same chunk. Now we want to do like a hierarchical clustering to see if there is a similarity between controlled wells or other treated wells, or if there is any anti-correlation. Before doing a similarity matrix, I would just go and to options and to display and link rows and columns. Why do I do that? I'm going to be sorting the columns based upon the compounds that they were treated with, based upon the mechanism of action. When we're doing that, we also want to link the rows and columns so that the sorting does not change the order. So I just linked rows and columns. And then now we can do the similarity matrix. I'll just make sure I just checked it. Yes. Once that's done, we can do a hierarchical clustering. And I'll be clustering on rows and columns. Then click OK. I would just zoom out for us to get an overall idea of the similarity matrix. So as you can see here, any big chunk of blue, whatever the measurements that are coming from these particular wells are like highly correlated, anti-correlated to these wells, or whatever the big chunk of red that you see here, we can say that the measurements coming out from these wells are highly correlated with whatever measurements coming out of the other well that is compared to. Again, I would definitely say this is like just a starting point for you to understand the biology, and it needs to be followed with with a lot of experiments to confirm what's exactly going on. But this is a good starting point to know if there is a high correlation or anti-correlation that is going on between your treatments. I would just zoom in so that you can see the details. And you can move around this way and see if your comp and okay, I didn't actually sort it before doing a hierarchical clustering. Now I can do it again to show once we sort the data, how it looks like once we sort based on compounds. So just let me quickly do it again. Sorry for the repetition. I'm just coloring based upon the different compounds. Or once that's done, I'm going to be linking those. sorry, I was doing a Pearson correlation based on similarity matrices, and then link rows. And now when you right click, oh, I'm sorry, when you click on the particular thing, now it is sorted based on plate, but I would like to sort based on compounds and MOA. So I would just go back and say compounds, and then followed by when you press shift and click MOA, then it is sorted both based on compound and MOA. So that's now sorted. Just making a quick shift. Yes. And now we can go back to my rampant clustering. Choose rows and columns. Yeah. Now this is like a zoomed in version, but you can see like all the wells that were treated with Aurora kinase inhibitors are like clustered together, and those that are treated with DNA replication are kind of clustered together. So this way you'll be able to see if any morphological profiles of compounds with unknown mechanism are clustering with the compounds with known mechanisms. So you can understand if it is doing something similar to whatever known compound of interest is doing. So I will go back, like zoom out, so that you can get a better idea of the similarity matrices. With this, I kind of come to the end of my demonstration. Please let me know if you have any questions. Drop it in the Zoom chat. I'm happy to answer. If not, we can proceed on to the hands-on exercise. Yes. I will just share the slide to give details about the workshop, and then we can break out, we can go into the breakout rooms to start working on it. Let me just give you an introduction to the data set that we are going to be using for the workshop. Do you see my screen, like slides? Yes. So the data set that we'll be working on today will contain images from MCF7 cancer cells, and the cancer cells were treated with 113 different compounds at 8 different concentration. And the image, the cells were stained for DNA, actin, and tubulin. And there's also a negative control DMSO. So I have added like 3 different data sets in the workshop material. You'll be having the feature selected data, or just the normalized data, or the data without normalization or feature selection. There's a written exercise which you can follow, and we need to load 3 different data for the exercises that we'll be doing. And it is like clearly mentioned in the written exercise, and there's also a screenshot PDF where you can kind of link from the written exercise to do all the changes that you need to make. And we'll see how the normalization or feature selection affects the similarity matrix. And with that, Beth Simony and other postdoctoral associates, Paula Llanos and Satovisha, will be helping you out in the breakout rooms. Feel free to ask any questions. And for more resources, or for understanding more about image-based profiles or anything that I've discussed today, I've posted all the links here. Feel free to check it out. And if you have any questions regarding your image analysis, feel free to post it on ImageSC forum. With that, I would like to thank Beth and all the members of Simony Lab. A special thanks to all the postdoctoral fellows who was very helpful for this workshop. And please do attend another cell profiler workshop from my team by Paula Llanos tomorrow on 3D segmentation.